What if I told you that your DNA isn't 100% human? Hidden inside the genome of every person are the remnants of ancient viruses that infected our ancestors millions of years ago. These forgotten viral fragments make up around 8% of the modern human genome, though most of us remain blissfully unaware of their existence. Yet far from being inert leftovers, many of these viral genetic elements play vital roles in human growth and development today. From the earliest stages of pregnancy to the health of our organs and tissues throughout life, these so-called endogenous retroviruses continue to influence our well-being in surprising ways. Some even regulate key aspects of the immune system and brain function. So how did these viruses become permanent residents of the human genome? Why weren't they eliminated by evolution over countless generations? And what impacts do they have on health and disease today? From their roles in embryonic development to their risks in organ transplantation, join me as we explore the secret viral passengers hidden within our DNA. Human endogenous retroviruses are viral sequences that have become permanent parts of the modern human genome over the course of evolution. They are estimated to make up around 8% of human DNA, a staggering amount. Considering our entire genome consists of about 3 billion DNA base pairs. HERVs originate from rare germline infections by exogenous retroviruses at various points in human evolution. Their viral genes were reverse transcribed into DNA and inserted into human chromosomes, allowing them to be passed down vertically from parent to offspring over generations. There they remain today as molecular fossils of those chance ancient infections. Structurally, HERVs resemble modern infectious retroviruses like HIV, but most HERV instances are incomplete or defective, having accumulated mutations that prevent them from forming infectious particles. While no longer able to replicate themselves, many HERV fragments retain the ability to be transcribed into RNA and even translated into proteins that carry out important functions. So rather than representing purely junk DNA, the majority of human endogenous retroviruses provide benefits to their hosts. However, some instances have been implicated in detrimental impacts as well. Ongoing research continues to uncover the diverse roles these viral genetic remnants play in human health and disease today. Human endogenous retroviruses play crucial roles in supporting embryonic and fetal development. Many HERV elements are specifically activated in early pregnancy, where they carry out vital functions. For example, the HERV-derived syncytin proteins fuse placental cells together into the syncytiotrophoblast layer that facilitates nutrient exchange between mother and fetus. By aiding the formation of this crucial placental structure, syncytins support fetal growth and development in the womb. Beyond fusion, HERVs also help maintain stem cell potency in embryonic structures during early development. This prevents premature differentiation while allowing organized growth. Additionally, HERVs enhance cell survival and prevent errors during rapid cell division in fetal tissues. So by assisting cell fusion, maintaining stemness, and enabling survival, human endogenous retroviruses support fetal growth at multiple key stages. Their activation facilitates proper viral replication in placental tissue, as well as overall bodily development in the growing embryo fetus. Further research continues to uncover additional HERV elements that contribute to early human development in diverse ways. While especially active during embryonic development, human endogenous retroviruses continue to influence human physiology long after birth as well. Many HERV elements regulate cell differentiation in organ and tissue development throughout life. For example, HERV-H expression guides differentiation of neurons and blood stem cells postnatally. HERV activity also modulates immune responses and inflammation pathways. Moreover, animal models indicate HERVs can mediate disease processes later in adulthood. In mice, certain endogenous retroviruses drive age-related conditions like leukemia arthritis, and autoimmunity when abnormally expressed. However, other models show protective antiviral effects instead. So the ultimate impacts of HER-Vs beyond birth appear complex and likely dependent on the specific element. Nonetheless, their continued transcriptional activity demonstrates these ancient viruses still help shape human developmental trajectories over the entire lifespan, not just during early pregnancy. Clarifying their diverse postnatal effects remains an active area of investigation today. The integration of ancient viruses into the human genome occurred over an extensive evolutionary timescale, spanning tens to hundreds of millions of years. Many HERV insertions likely took place in early mammalian ancestors, as similar viral sequences exist in other mammals, like mice and chimpanzees. The origins of HERVs thus trace back over 200 million years to the divergence of mammals. Continued germline infections introduced new viral variants into various lineages over subsequent eons. More recent integrations occurred since the emergence of the Homo genus 5-7 million years ago. These newer viral remnants tend to be more intact and active. They also pose greater risks of detrimental impacts compared to older, more mutated HERVs. For example, HERV-K insertions began infiltrating hominin genomes just a few million years ago and remain transcriptionally active. Such recent integrations seeded existing HERV families with new members that retained functionality. Their introduction coincided with crucial junctures in human evolution, like the development of a large brain. So endogenous retroviruses chronicle a long history of viral interactions, spanning both deep evolutionary timescales and more recent human emergence. This complex timeline underlies modern HERV diversity and its multifaceted impacts on human health today. In addition to their developmental roles, some human endogenous retroviruses influence disease processes later in life. Their effects appear complex. While some HERV activities cause illness, others prevent it. For example, the HERV-derived hemoprotein offers protection during pregnancy. By guarding the placenta, hemo facilitates healthy fetal development and prevents complications like preeclampsia. So this viral gene fragment provides a clear benefit to the mother and baby. In contrast, increased expression of the HERV-W and HERV-K families has been linked to multiple sclerosis, MS, and schizophrenia, respectively. While not definitive causes, their abnormal activity correlates to inflammation and neurological dysfunction. So these HERVs may worsen disease processes in certain contexts. The ultimate impacts likely depend on the specific HERV, its location in the genome, expression levels, and other interacting genes. But the influence of these ancient viruses on modern human disease is undeniable. In simple terms, some HERV elements help prevent illness, while others contribute to it. Disentangling their precise effects remains an ongoing aim of medical research, with implications for better understanding complex conditions. Continued investigation of endogenous retroviruses will clarify how to leverage their benefits while minimizing potential harms. Gene editing technologies like CRISPR could potentially be used to remove or alter harmful human endogenous retrovirus (HERV) expressions while preserving beneficial ones. CRISPR allows for precise editing of DNA sequences. Researchers have already used it in cell and animal models to eliminate activity of specific endogenous retroviruses. Similar approaches could remove disease-linked HERV insertions from human genomes or disrupt their ability to be transcribed. However, gene editing in humans does carry ethical concerns and technical challenges. Safety risks like off-target effects must be minimized, especially for any somatic cell. Gene therapy aimed at altering HERV activity levels. Germline editing to completely eliminate harmful variants from our species' gene pool would be even more controversial. Nonetheless, the specificity of CRISPR makes it well suited for selectively editing endogenous retroviral genetic remnants based on their contributions to health. With further advancement and prudent application, these technologies could help elucidate and leverage HERV benefits while mitigating their detrimental impacts. Looking ahead, human endogenous retroviruses may not be a closed chapter in human evolution. Some evidence suggests we could acquire new genomic integrations in the future as well. HERVs share many functional similarities with modern infectious retroviruses like HIV. Since HIV inserts its genome into host DNA as part of its replication, it demonstrates that exogenous retroviral integration remains possible in present-day humans. Thus, given the right circumstances, humans may yet again face germline infiltrations, leading to new endogenous retroviral variants. Any such integrations would introduce new genetic fragments with unknown impacts for future generations. While speculative, this raises intriguing questions about viral contributions to not just our past, but also our future evolution as a species. Extensive interplay exists between viruses and human physiology over evolutionary timescales. Endogenous retroviral sequences chronicle a long history of chance infections, with the majority providing benefits after integration. Yet some HERV activities also detrimentally contribute to complex conditions like autoimmunity, cancer, and neurological disease. Further research should continue elucidating their diverse effects on prenatal development, lifelong health trajectories, and even potential future evolution.